Namaskar and welcome to the TY BSc students in this third lecture about the Mendelian genetics, that is Mendelism. I hope in previous lectures you have get familiar with all the laws which are stated by the Mendel, and in this lecture we will study the interaction between the genes. From these funny pictures you will get idea. I think you have get idea about the gene interaction. So let us see what are the different types of the genes interactions we are going to see in the next slide. From this subunit we will get idea, or we can say we can see that what type of different advancements are happen after the Mendelian study about the genetics. So let us see the introduction about this gene interaction. That is, first of all, the Mendelian genetics, which does not explain all kinds of the inheritance, for which the phenotypic ratios in some cases are different from the Mendelian ratios. So regarding this point we will see the different ratios which are occurred after the advanced studies in the genetics. So which type of different ratios are happen, that will we see also in the next slides. This is because the sometimes a particular allele may be partially or equally dominant to the other, or due to existence of more than two alleles, or due to lethal alleles. Lethal means toxic or killing alleles. These kinds of genetic interactions between the alleles of a single gene are referred to as allelic or intra-allelic interactions. Non-allelic or inter-allelic interactions also occur where the development of single character is due to two or more genes affecting the gene expression of each other in various ways. Thus, the expression of gene is not independent of each other and dependent on the presence or absence of the other gene or genes. These kinds of deviations from Mendelian one gene, one trait concept is known as factor hypothesis or interaction of the genes. So I think you have get proper idea about this gene interaction. In previous lectures we have seen different Mendelian laws. There are particular fixed type of ratios occurred, but after the advanced studies in the genetics, the gene interactions, that is two different types, that is allelic and non-allelic gene interactions, shows the ratios different from the Mendelian ratios of which we have seen in the previous lectures about when we get understood about the different laws of the Mendel's. In this lecture we will study the different types of allelic gene interactions with the help of different examples. what type of ratios occurred in the allelic gene interactions. So first type that is first type in the allelic gene interaction is incomplete dominance or blending inheritance, which shows the ratio 1 as to 2 as to 1. So this incomplete dominance, in which a dominant allele may not completely suppress other allele. Hence, a heterozygote is phenotypically distinguishable or intermediate phenotype from the either homozygote. In this, we have taken the example of flower color in the 4 o'clock plant, in which the ratio of monohybrid cross is occurred, that is 1 as to 2 as to 1, and in which we are going to see this in this picture, that the cross between red colored 4 o'clock plant and white colored flowered 4 o'clock plant, the F1 generation shows the pink colored flowers, that is, it shows the allelic interaction shows that the intermediate phenotype, that is uh, which cannot completely suppresses other allele. Here we can see the pink colored is occurred in the F1 generation, and by selfing the F1 generation with itself, the checkerboard shows the one red colored individual red flowered individual occurs, one white flowered individual occurs, and two pink colored individual flowered individuals occurs, which shows the ratio 1 as to 2 as to 1, which shows the incomplete dominance of the red colored flowered plant over the white flowered 4 o'clock plant. Type is codominance. Here, both the alleles of a gene express themselves in the heterozygote. Phenotypes of both the parents appear in the F1 hybrid rather than the intermediate phenotype. In this, we have taken the example of human Mn blood group is controlled by single gene. For this purpose, or for better understanding of codominance, I will provide a particular link of detailed explanation about the codominance in the description box, or in the uh, or at the end of the video. Otherwise, this regarding the codominance explanation or the blood group explanation, the video will be more lengthy. So that for that purpose, I have given the link in the regarding this codominance. The third type, that is overdominance. In this, sometimes the phenotype of F1 heterozygote is more extreme than that of either parent. So in this, the example is the regarding the Drosophila melanogaster, that is, in which the amount of fluorescent eye pigment in the heterozygous white eyes of the Drosophila exceeds than that of found in either parent. So regarding this also, I have provided the video which properly explains how the fluorescent red colored eyes Drosophila get over dominance on the white-eyed Drosophila. So please see the video regarding this over dominance, which regarding which the link is provided. Fourth important factor, or we can say which is very dangerous factor, that is lethal factor, which shows the ratio 2:1. So regarding this also I have provided a simple video which shows the proper explanation about this poisonous factor or lethal factor. The genes which cause the death of the individual carrying it is called a lethal factor. Recessive lethals are expressed only when they are in homozygous state and the heterozygotes remain unaffected. Regarding this, the example is yellow coat color in mice, which is properly explained in the video. So please see the video. And the last important type in the allelic gene interaction is multiple alleles, in which a gene for a particular character may have more than two allelomorphs or alleles occupying same locus of the chromosome. So in which uh, particularly in the allelic, these allelomorphs make a series of multiple alleles. So in this multiple alleles, occurrence of more than two alleles in a single locus, the examples are ABO blood group system in humans and self sterility in the tobacco. This is all about the gene interactions. Now we welcome our newly admitted member, that is Miss Rajeshwari Taugule. Already our head department of botany, Dr. Priyanka Jadhav, had welcomed you. So on behalf of our department, I again welcome you, Miss Rajeshwari Taugule. Regarding my lectures or previous lectures and all the notes which I have provided earlier, I will provide you in the next lecture very soon. So this is all about the today's lecture about the gene interaction. In next lecture, we will see the remaining part, that is non-allelic gene interaction. So thank you. Thanks one and all to the TYBSc students.